Hey guys, welcome and welcome back to my channel. This is Dr. Prasanjit Das, currently JR1 in the Department of Pharmacology, AIMS Kalyani, and I am back with my previous seminar. So this is the seminar which I have already covered in one of my previous videos, but this is a detailed video of the previous seminar. So without wasting any time further, let's get into today's video. In today's video, I am going to cover the topic that is bioinformatics tools in pharmacology. So I have already taken this class in one of my previous videos, but this is a better version, that is a more detailed version of the same topic. So the topics of discussion in this particular seminar will be the introduction to bioinformatics and the history of bioinformatics, then bioinformatics tools classification, then the goals and field of bioinformatics, then we have the applications of bioinformatics, and finally the limitations of bioinformatics, following which I will be providing you with one conclusion. So first, coming to the introduction. So bioinformatics, it is basically a union of biology and informatics. So in bioinformatics, what we do is we store, then we retrieve, then we analyze and manipulate the biological data, that is the data which is related to several biomolecules, that is macromolecules like DNA, RNA, proteins, then several metabolites. With this data, we can get several information about the particular molecules, and we can also analyze the data. So for this, the only thing which we need is a computer and a stable internet connection, because most of the bioinformatics software are online and they are also free to use. So here you can see the diagram, that is bioinformatics is basically the combination of medicine, biology, maths and physics and computer science. So that is, it integrates the biological data with the computer sciences, that is with the help of several statistical and mathematical equations, it helps to derive conclusions from the biological data. The other diagram is also the similar thing, that is it represents the use of computer science for analyzing the data which is obtained from several specimens, that is biological specimens. Then we can also do some analysis of the compounds, their structure, etc. So this is about bioinformatics. Now coming to the history of bioinformatics. So in the year 1960, protein sequence data was managed using high level computer language, and Margaret O. Day-Off, that is, she was considered as the founder of bioinformatics, that is the pioneer in bioinformatics, because she was able to compile all this protein sequence together, and then she created a protein and a nucleic acid database, and also the tools so as to retrieve this information from the databases. That if we want a particular information that is regarding any particular protein or nucleic acid, then we can retrieve it from the database. And she also published this work in one of her books, that is Atlas of Protein Sequence and Structure. So this is the reason why she is called as the founder of bioinformatics, and this was about the history of bioinformatics. Now coming to the different types of bioinformatic tools, that is databases and the classification. So we can classify it into several databases, and the method of classification also varies. So first method of classification is classifying the databases into primary database and secondary database and composite databases. So primary databases. So primary databases basically contain the original information which we obtain from the experiments. So it will include original information like the DNA sequence and the protein sequence, that is amino acid sequence in the proteins and the different protein sequence in a particular molecule. Then secondary databases, these are the databases which obtain the information from primary database, and then they analyze it, and then they form a particular database. So in this we have several examples like mutation analysis database. So in this, basically the data sequence which is present in the primary database, that is basically the DNA, in that we will look for several mutations, and with that we will create a database that is known as the mutation analysis database, which is an example of secondary database. Now next is composite database. So composite database is nothing but a combination of several primary databases. So in this way we will be saving time and we will be avoiding ourselves searching in several different sources. So we will search in one particular source and we can access several primary sources in one complete database. So one such example is NCBI, that is National Center for Biotechnology Information. It hosts several databases, that is the primary databases basically. So this is about the composite database, secondary database, and the primary database. So this was one method of classification of the bioinformatic tools and database. Now coming to the next method of classification, that is classifying the biological database based on the nature of information they contain. So either the database can contain some sequence. So in this we can have several databases like protein databases and nucleic acid databases. One such example is UniProt. So it is a database which contains the information about the proteins and the amino acids. Next is another type of database, that is macromolecular 3D structure database. In this we will get the information about the structure of different compounds. So for example, we can have examples like nucleotide database, because in this we will see the structure of the nucleotide and we will also see the protein structure database. So one such example is the protein data bank, which is an example of macromolecular 3D structure database. So I will be discussing about these two databases in detail in the upcoming slides also. Now coming to the final method of classification, that is we can classify the bioinformatics tools or databases into protein databases, then we can classify it into disease databases, then metabolite databases, then sequence databases, and pathway databases. So in protein databases, it is simple from its name that we will be understanding about the different protein structure, then the amino acid sequence, then the protein interactions also can be seen. Then in disease database, we will look for different diseases. Metabolite database, we will look for several metabolites, small metabolites, and the metabolic pathway. Then we will also look for the sequence, that is DNA sequence. Then we have the RNA composition, then we have the protein composition. All those things can be seen by the sequence database. And finally coming to the pathway database, that is it is helpful in finding out the interaction between two different molecules in a particular organism or in the human body. Now coming to the examples of each of these types of databases. First is protein database. So in protein database we have first, that is PDB, that is protein data bank. So in this we can get the 3D structure model of the particular proteins. And then we have UniProt. It is also a protein database in which we get the amino acid sequence and the protein sequence of a particular molecule. Next is OMIM. So OMIM stands for Online Mendelian Inheritance in Man. So in this we are actually studying the different types of genetic diseases and the genotypes which are associated to that particular phenotype, that is the genes which are responsible for causing particular diseases. So it is a type of disease database. So these two were types of protein database. This is the type of disease database. Then coming to next two databases, that is HMDB and ECMDB. So these two are examples of metabolite database. That is HMDB is human metabolome database. So in this we will be studying about the human metabolites, and ECMDB is basically E. coli metabolome database. So here we will study the metabolites which are present in the E. coli bacteria. Now coming to the next type of database, that is the sequence databases. So in this we have examples like GenBank, then we have BLAST, that is basic local alignment search tool. So in this we are actually looking into the nucleotide sequence in case of GenBank, and in case of the BLAST, that is basic local alignment search tool, in that we can also look for proteins, we can also look for the nucleotides. So these two are examples of sequence database. We will be discussing each of this in the upcoming slides. Then next is DDBJ, that is DNA Data Bank of Japan. It is also a sequence database, that is it comprises of the DNA sequences. Then we have Ensemble, which is also an example of sequence database. Here we get the different genomes of vertebrates. So basically it is a collection of genomes from different vertebrates including human beings and other vertebrates. Next is KEGG pathway database. So this is a pathway database and it stands for Kyoto Encyclopedia of Genes and Genomics. So in this we will find information regarding several pathways. Then next is BioGrid. BioGrid, it is also a pathway database, and here we can find information about the post-translational modifications in the protein. Then we can also find about the several interaction between different proteins. Then MINT, it is also a pathway database. So MINT stands for Molecular Interaction Database. So here also we can see several sorts of interaction, and most of these databases are free to use. And actually not most of this, almost all the databases which I have mentioned so far are free to use. And coming to one important point regarding the BLAST, that is basic local alignment search tool. So in this we can actually give a query sequence, like a query sequence of nucleotide or protein, something which we found out from experiment, and then we can compare it with the proteins sequence or the nucleotide sequence which is present in the database. So what happens after the entry of the query sequence is that it gets aligned with the sequences which are already present in the database, and the sequence to which it is aligning the maximum, that sequence is considered as the homologue, and as a result now we can predict the sequence of the query DNA or the query RNA or the query nucleic acid or the query protein. So this is helpful for finding and sequencing the different genetic material and the proteins. So this is a very useful tool, and this is one of the easiest and simplest tools out of all these bioinformatics tools. Now coming to the final one, that is BioInform. So this is basically providing information about the different bioinformatics technology which is upcoming, and it gives the updates regarding the different bioinformatic tools. And as I have already mentioned, most of the databases are free of charge, and some of these databases can also charge a nominal fee. That is basically they are subscription based also. So if you subscribe to them, then you get the complete access to those databases. So first is the UniProt database. So first we will search UniProt in Google, and then we'll click on this UniProt. So UniProt is basically for searching about the proteins. So in this search bar we can type our desired search. So like for example, we can type insulin here and then click on enter. So in this you will see for insulin we are getting several results, and this symbol means that it is verified by Swissprot. So it means it is manually annotated, whereas the other one, that is Tremble, it is not manually annotated yet, and its status is still pending, like it has to be reviewed again. So suppose if we are clicking on this one, that is INSR human insulin receptor. So in this we have the entry number as P06213. So we will click on this, and then we will get all the information regarding INSR underscore human. So basically it is the insulin protein. So we will get the function of the proteins, then different taxonomical classification of the proteins, then the gene names, then the organism, since it is human, so it is Homo sapiens. Then we can get the subcellular location of the particular query, for example, where it is present. Then after that we can also look for the diseases associated and the variants. So here you can see the description, that is severe insulin resistance syndrome characterized by insulin resistant diabetes mellitus with pineal hyperplasia and somatic abnormalities. So this is the description for this Rapson-Mendenhall syndrome, in which there is an involvement of insulin. So this was about UniProt, that is a database for protein. Now coming to the next one, that is OMIM. So OMIM stands for Online Mendelian Inheritance in Man. So it is for diseases. So we'll click on OMIM. Then we can search for any particular disease. So we can search here any diseases, like suppose we are searching diabetes. So then it will give us the following results in which diabetes is present. So suppose if we want to see this particular hit, that is type 2 diabetes mellitus T2D, and its number is 125853, that is the entry number. Click on this, then we can get all the details regarding type 2 diabetes mellitus, that is the description, then the inheritance, how it is being inherited, and along with that we will also get the references. So if we click on this reference and if you open it in a separate tab, then you can get the article in which this particular text is being quoted. Then we can also see the biochemical features regarding that particular disease, the molecular genetics related to that particular disease, clinical management, then pathogenesis. And for every database, like and for every information there is actually reference which you can click and directly access that. And Also in a OM database, we can get external links. So here you can see it is related to several other databases also. So if you click on clinical trials, it will show you the data which is related to diabetes mellitus and clinical trials. That is clinical trials in diabetes mellitus, then gene reviews, genetic alliance, then Medline plus genetics. So with this, you can get the entire information about a particular disease. So this was about OMIM database. Then coming to the next one, that is HMDB, that is the Human Metabolome Database. It is a metabolic database, that is a database for metabolites. So we'll click on it. So this is the interface. So in this, we will select metabolites. So it is already selected as a metabolite. Then we can search any of the metabolites. So here I will search for acrolein. So then we click enter, and then we can get all the different hits. So it is displaying 17 metabolites. So basically these are all the metabolites which include acrolein in their description. So first is acrolein itself, then we have trans 2-pentanol, which is in which they have variations of acrolein. That is, with acrolein they are having different functional groups, and corresponding to each name you will get their corresponding ID, and you will also get their structure. So if you want to see a particular molecule or a particular metabolite like acrolein, then you will click on this acrolein, and then it is loading. Now we can get the description of acrolein, then we can get the structure, and it can be downloaded also in different formats. So then we can also get the chemical formula for that particular metabolite, the molecular weight of that particular metabolite, then IUPAC name also. Then IUPAC name and traditional name is the one which we know. Then also the registry number. Then it belongs to which kind of compounds, that is organic oxygen compounds. Then if we click on it and if we move on to a separate tab, then we can also get details about that also. Then its different effects, that is physiological effects. So it can cause several health effects like bleeding, as we already know that acrolein is implicated in hemorrhagic cystitis. Then it is also responsible for causing vomiting, uremia. Now coming to the next database, that is GenBank. So GenBank is basically meant for seeing the nucleotide sequence. So here we can see, we can select nucleotide, and then we can search for any of the compounds, and we can get the nucleotide sequence for that particular compound. For example, if we search here insulin, or else let's search a different molecule like glucagon, and then we press enter. So in that case we will get the different hits regarding insulin. So here species wise it is being classified: plants, fungi, bacteria, and all those things. Then the different nucleotide sequence. Now here if we click on the fourth one, that is human glucagon mRNA. So it will tell us about the human glucagon mRNA. So here it is the description: organism, the accession number, that is the identity number of this particular entry, then the features, and the rest of the details are also present, plus the nucleotide sequence which is responsible for coding this glucagon in humans. So this is about the nucleotides which are present for glucagon. And we can also run blast is also another database. Next I will be discussing about blast also. So from this interface also we can run blast. Now coming to the next one, that is the blast database, that is basic local alignment search tool. So in this we can look for the nucleotide sequence also, and we can also look for the protein sequences. So we give a query sequence, that is the sequence which we are having, and then we match it with the sequences which are present in this database. So let us take an example of the like nucleotide sequencing. So we will click on nucleotide blast. Then in this we can provide the accession number, or we can provide the faster sequence, which is nothing but the nucleotide sequence which we are having. And we can also upload a file like a Word document in which we are having the nucleotide sequence of that particular genetic material which we are having. Then we can provide running title for this. Then we can select nucleotide collection, which is already being selected here, and after providing the details we can click on blast. So then we will see the alignment, that is if our particular sequence is matching with any of the nucleotide sequences which is present in this BLAST database. Now coming to the final one, that is the metabolic pathway. So it is like a pathway database. So that is KEG. So KEG pathway database stands for Kyoto Encyclopedia of Genes and Genomes. So in this we can look for different metabolic pathways. So when we scroll down, we can see several metabolic pathways. So suppose we take carbohydrate metabolism, under which we select galactose metabolism, that is we want to see the different enzymes and the substrates which are present in galactose metabolism. So we will click on galactose metabolism, and then it will be opening a complete flow diagram for the galactose metabolism. So here you can see the different substrates and the products, and in between the boxes which contains the number is actually the enzyme. So if you want to know about that particular enzyme which is responsible for this conversion, then we will just click on this number, and then we will get the name of the enzyme. So that particular enzyme which was coded as 3.1.1.25 is actually 1,4-lactonase. Now coming to the goals of bioinformatics. So the principal aim of bioinformatics is to understand the living cell and the life processes. So basically it understands the central dogma of life. So that is, central dogma is basically the DNA converting to DNA itself, that is replication, then DNA converting to RNA, that is transcription, then RNA completely converting into protein, that is known as translation, and this protein is the one which is responsible for mediating the several physiological effects of the cell. So by understanding this molecular sequence and the structural data of the DNA, RNA, and the proteins, we are actually understanding the activities of the cell and the physiological effects which are mediated by these molecules. So this is the main goal of bioinformatics. Now coming to the field of bioinformatics, bioinformatic tools are used in three different types of analysis, that is molecular sequence analysis, then molecular structure analysis, then molecular functional analysis. Coming to molecular sequence analysis, so in this first one is sequence alignment, as we have already mentioned in BLAST, that is basic local alignment search tool. In that the query sequence gets aligned with the sequences which are present in the database already. Then next is sequence data based searching. So basically we are searching for different databases and the different sequencing which is present in the databases. Now the next thing is finding the gene and the promoter, and then we can also find the motifs, that is the repetitive sequence which are present in the DNA, and it can alter the function of the genetic material also. Next is molecular structure analysis. Under this we are having protein and nucleic acid structure analysis, then comparison of the different protein structures, then with the help of that classification of the different proteins and the nucleic acids, and finally doing the prediction of different proteins by the protein models which are already present in the databases. Next is molecular functional analysis, that is it comprises of gene expression profiling and protein-protein interaction prediction. Also it comprises of the simulation of the metabolic pathway. So in this we will have the KEG pathway database, that is meant for the elucidating the different pathways. Now coming to the applications of bioinformatics, so here you can see there are several applications of bioinformatics, that is it is used in personalized medicine, that is individualizing the healthcare. Then we can use it for forensic analysis, and we can use it in pharmacogenomics, that is gene directed therapy. Then we can also use it for discovery of drugs, we can also use it for phylogenetic analysis, you can also use it for agriculture technology development, and also for protein ligand interaction analysis. So these are the several applications of bioinformatics. Now coming to each of this individually, first is the drug discovery. So here you can see we have the chromosomes. So the chromosome contains the gene, and the gene codes for amino acids, and this amino acids then finally folds to form the protein, and this protein is having a 3D structure, and this 3D structure is responsible for its function, and this function is responsible for carrying out several life processes in the cell. And these life processes, when understood, we can actually devise several drugs which can act on this life process. So if we know the normal process, and if we know the pathological process which is occurring in a disease condition, then we can give a drug in such a way that it alters the pathological process. So this is the way how drugs are discovered with the help of bioinformatics, because bioinformatics come into play in almost every step of this, that is in gene analysis, then in amino acid analysis, in protein sequencing, then in the structure analysis, then in the functional analysis of the proteins. Then one such example where bioinformatics have played a great role in the development of new drugs is the development of imatinib mesylate. So this is a drug which is used for the management of chronic myeloid leukemia, that is it inhibits the formation of the defective protein of CML, that is the defective protein which is seen in chronic myeloid leukemia. And when we do the analysis of proteins and we compare the proteins of a normal individual to that of a pathological state, then that is what we call as proteomic analysis. And if we use gel electrophoresis in that proteomic analysis, then it is called as gel electrophoresis proteomic analysis. So proteomic analysis. So in gel electrophoresis, what we get is we get high resolution images, that is 2D images, and these are actually stored in a database which is also known as World 2D Page Repository. So in this we get the high resolution 2D images of the gel electrophoresis which is done for different proteins. So this is one application of bioinformatics in drug discovery. Now continuing with that, also bioinformatics is used in the several steps of the development of the drug. So we know that the development of drug begins from target discovery, then it goes to drug discovery, then finally to the drug development. So in each of these steps we have bioinformatics involved. So in target discovery, that is target identification and target validation, we are actually finding the structure and the molecules which are binding to that particular target. So we are doing the molecular docking. So in this also we are having the role of bioinformatics, that is the proteomics, transcriptomics. Then we have the homology modeling, that is we are thinking and we are finding out the structure of the protein by the amino acid sequences. Then in drug discovery phase also, that is where we are identifying the lead and we are optimizing the lead. There also we need bioinformatics. And finally in preclinical and clinical trials, there we need bioinformatics for assessing the toxicity profile, then for assessing the several biomarkers which are showing that the drug is particularly working on a particular condition. So in all the cases we need bioinformatics. And when we incorporate the data, that is from the genes, that is genomics, from the proteins, that is proteomics, and from the mRNA, that is transcriptomics, to the clinical data which we obtain from the experiments and the trials, we can establish a proper relationship between a particular effect and the drug, and what are the factors which are actually responsible for causing that particular effect. So with this we can help in discovering the drug in an efficient manner. Next is, by bioinformatics we can also find the molecules which are very much similar in the pathogen and also in the human beings. So as you can see that these two molecules are almost similar, that is the pathogen target molecule and the molecule which is of the host, that is human. So if we use bioinformatics and we can find out one minor change, as you can see here, this is the minor change between these two, then in that case we can develop some drugs which are specifically acting on this minor change and not on these minor change. So in this case we can develop the drugs which are specifically acting on the pathogen target and not acting on the host cell, leading to decrease in the adverse effects which was earlier produced by the drugs which acted on the same molecule, unlike the present drug which is acting on the minor change which is present in these two molecules. So this is also one example where bioinformatics play a major role in the development of the drug. Continuing with the drug discovery, one of the earliest contributions of bioinformatics in the drug target discovery was the identification of the sequence homology between the simian sarcoma virus oncogene V6 and the platelet derived growth factor. So we know the structure of the platelet derived growth factor, and as a result it helped us in discovering the drugs which can act against this particular virus also. So this is also an example where bioinformatics play a major role in the development of new drugs. Now coming to the flowchart of in silico approach for drug designing. So in this we have the protein data bank and we have the homology modeling. So in homology modeling, what is done is basically we think of the 3D structure of the protein by the amino acid sequences, and the protein data bank provides us the access to different types of proteins and their structure. So this is being stored in the chemical libraries, and with the help of that we are doing virtual screening, that is we are doing molecular docking, that is we are trying to find the molecules which are actually fitting to target site of the molecule, which is thought to mediate a better effect. So then we do the process of molecular docking, in which we are trying to fit one molecule into the enzymes active site or the protein's active site, and then we do QSAR, that is quantitative structure activity relationship, that is it is basically analyzing the biological effect by the physiochemical property of this interaction between two molecules in molecular docking. And finally, what we do is we do the pharmacophore modeling, that is with the help of this we find out several other molecules which are having the similar structure, and as a result this helps in hastening the process of the development of the drugs. And finally we look for the absorption, distribution, metabolism and excretion profile of the drugs with the help of some of the databases like Swiss ADME. Now coming to the next application, that is personalized medicine. That is the patient's genome can be sequenced very fast with the present technologies, and as a result we can find out the harmful mutations which are present in the particular gene, and this can actually predispose the individual to several diseases. So we can do early diagnosis, and hence we can do the effective treatment also. And then application of bioinformatics is also seen in case of cancer therapy, because in cancer therapy there are several cells. The cancer cells are basically having several mutations, so if we find out these mutations we can devise the drugs against that particular mutation also. So this is the way in which the bioinformatics play a major role in the treatment of cancers. Like one example is the one which I have already explained, that is imatinib for the treatment of chronic myeloid leukemia, that is it prevents the formation of the defective protein which is seen in case of chronic myeloid leukemia. Then other applications is in polymorphism research. So single nucleotide polymorphism, that is the change in a one particular nucleotide which is seen, is called a single nucleotide polymorphism, and this is actually predisposing some of the individuals to several diseases like diabetes mellitus and hypertension. So if we detect it earlier, then we can also take the necessary intervention so as to prevent and treat those diseases. Now coming to the next one, that is the application of bioinformatics in forensic medicine. So the phylogenetic analysis reports can be used as an evidence in the criminal courts. So this is the application of bioinformatics in forensic medicine. Then in case of agriculture advancement, the plant genome is stored in databases, and it helps us to develop new varieties of crops which are actually having good resistance compared to the other crops, and are also having greater productivity. So this helps in the development of good variety of crops. Next is in the development of the antimicrobial drugs and also to prevent antimicrobial resistance. So bioinformatic analysis revealed a glutamate transport system which is present in the pathogen, that is Clostridium perfringens, but it is absent in case of mammals and birds. So if we develop a drug which acts against this glutamate transport system, then it will be selectively killing Clostridium perfringens, but it won't kill the mammals and birds, which includes humans and also the other mammals like other mammals and fowls. So this is one such application of bioinformatics for the development of antimicrobial drugs. Then another example is of Giardia intestinalis. So it comprises of a pathway, that is PI3K pathway, that is phosphoinositide 3K pathway, which leads to the formation of several proteins. So this pathway which produces some proteins is also present in in humans also, but the proteins which are being produced by this pathway in case of Giardia intestinalis is having a specific insertion, basically a specific amino acid due to a specific insertion in the genetic sequence. So if we target the drug against this specific genetic sequence, then we can kill specifically the Giardia intestinalis and not affecting the human cells. Then finally coming to the limitations of bioinformatics. So bioinformatics has got a number of inherent limitations. Those are the following. That is, first is high quality data. So high quality data is required for analysis. So if we get low quality data in case of any bioinformatics research, then it cannot be used further for getting meaningful results out of it. Next is, bioinformatics can never replace the traditional experiments which we use for testing the hypothesis. Thirdly, the sequence data which we obtain from high throughput analysis can obviously contain some of the errors, and if it contains the errors, then what we do is, like, if we do the downstream analysis of the data which is obtained from this high throughput analysis, then those data will be also having errors, so it leads to an increase in the chance of errors. These three, that is, requirement of high quality data, then next is it cannot be used for traditional experimental research methods for testing hypothesis, then third, the sequence data, if it contains errors, then it would lead to error containing downstream analysis results. So these are the disadvantages of bioinformatics in a broader way. Now finally, coming to the conclusion, bioinformatics is actually applying in silico analysis, that is using computer and internet different softwares to analyze and store, manipulate, and also to retrieve the information, and so as to design new products. And bioinformatic tools also detects the association between several genotypic and phenotypic data, as you can see here, that is the expression of the different genotypes and its resulting phenotype can be also found out with the help of bioinformatics tools. So these are the two major aspects of bioinformatics, and it has helped in translational science, that is, it has helped to apply the results which we obtain from several experiments into the improvement of the healthcare and also develop personalized medicine. So these are the references which I have referred for this presentation, and with this I would end the presentation. So thank you guys. So if you like my content, please do like, share, subscribe, comment, and also press the bell icon so as to get notified whenever I upload a new video. Thank you guys.